Very good afternoon to everybody. I warmly welcome you all in my class. I hope you all are doing very well. And in this particular class session, as you all quite aware about the fact that we have been discussing about the various aspects of environmental biotechnology. And to be very honest, that environmental biotechnology is a particular type of dimension where we have been discussing about different aspects which are related to the various achievements in the modern science. And we can actually implicate all these achievements or all the various kinds of technological advancement for the sake of human welfare. So basically, the conception of biotechnology is to develop the humankind in order to have a sustainable lifestyle, in order to improve the values of human lifestyle. And here in this particular case, we are using different strategy. We are using different kind of, the knowledge in the direction of modern science and technology. And basically, we are just trying to improve the lifestyle of our modern civilization. Now, the thing is that we consider ourselves that we have grown up and we have been in the way or in the track of civilization. And in this due course, we have upgraded ourselves to a great extent. And we are quite appreciating the fact that we have got some ideas, we have got some modern technology, we are very much, upgraded based on the industrial revolution and so on. But the problem comes, or something which always plays in our mind, that is the global issues, or some of the global issues which are raising the problem, like environmental degradation. So we forgot about the sustainability of our environment whenever we have concentrated towards the particular urbanization and industrialization. Now, the urbanization and industrialization, these two things are the, two important integral part of our civilization. That is important, and we should also, we are considering these two important part of our civilization, that urbanization and the industrialization. Without these two things, we cannot move or we cannot think a single day. But at the same time, we have to think for the environment. We have forgot that our environment is now under a great challenge. We have done something which is not good for our environment, because in the present days we are facing huge problems like climate change. Climate change is a global issue, and scientific personnel in every sphere of the globe, they are nowadays thinking about the remedy, or they are trying to take the precautionary measures in order to combat with the great problem, that is the environmental pollution, or the, in a broad aspect, the climate change, which is a great problem. Now, whenever we are talking about the climate change, we have already, we all are quite familiar with the fact that global warming is a very important phenomenon, and the global warming is caused not in a single day. In a gradual change in environmental pattern, or the gradual change in the environmental condition, give rise to this particular problem, that is the global warming. And we all are it is known to everybody that global warming is causing the problem which is known as glacier melting also. Now, the glacial melting, as a result, it causes the great problem like sea level rise. We know that the sea level rise is a problem which actually will hit our environment or our different biosphere component of our earth. Now, as per the prediction by the, the particular Environmental Protection Agency, they have speculated that within, a few decades, like the one of the heritage of the, in India, that is the Sundarban area, will be, under the sea. So this prediction is driven by the some of the simulation studies and the based on the pattern of the climate change. Now these are the great problems, and we are actually facing the problem nowadays. People are thinking about the environment. That's why in every course curriculum environmental science is included, so that the public awareness should be there in or the public awareness should be kept in mind in every time. Otherwise we will fail to keep our environment in a proper way. So whenever we are talking about the environment, we must remember that our environment is in a great threat. So we in order to overcome all these various kinds of hurdles or the different kind of raised challenges, we have to take some precautionary measures. Or whenever we are observing that already the environment is contaminated, or already the environment is polluted with some of the pollutants which are not good for our environment and may create some problems which are associated to our health. And so health associated issues are also very important. That's why the public health engineer, these people are very much concerned nowadays that what would be happen if we cannot control this kind of situation. That's why we need to clean our environment, and the environmental cleanup, this particular perception is coming and playing all the time in our mind, how to handle this situation or how to overcome this challenging situation. So in this direction, we can implement the biotechnology can be very promising and can be one of the great arsenal in order to combat with this great challenge. And environmental cleanup or environmental remediation is a very good way to combat with this problem. Now, in this direction, we can implement the concept of biotechnology, and we can implement the concept of biotechnology to remediate our environment. So the concept bioremediation comes in this particular case. So we can actually improve our environment, we can clean up our environment using the concept biotechnology. Now we are trying to involve ourselves in the direction of bioremediation so that we can get rid of the great problem that is the environmental pollution. Since last few classes, we have been discussing about the environment. We have been discussing about the various aspects of environments, why environmental science is important, why should we study environmental science, as well as why should we study about the different components of our environment like microorganisms. Every time we used to say the microorganisms are the culprit and they are creating different kind of diseases, and they are basically creating harm in the society. But it's not the true fact. If we know and if we understand microorganism properly, then we can also implement microorganism, or we can actually apply microorganism for the sake of human welfare. We can implement, we can understand the blessings of microorganisms, so we can improve the technology, or we can use the microorganism for the beneficial effect of the microorganism in order to have the betterment of our civilization. So we have concentrated on the microbiology also. We have studied a bit about the environmental microbiology. What are the different types of microorganisms are present in our environment and that are contributing a lot in order to combat with several kind of challenges, in order to improve the livelihood of the humankind. So in every aspect or in every way, we can actually implicate the blessings of microorganisms. We can conceptualize microorganisms. We can also find out those microorganisms which are unknown. Basically, last day we have been discussing about those microbial consortia or the microbial populations which are present in our nature, but we don't know about those microorganisms. So in order to unveil those microorganisms, or in order to understand those microorganisms, we can actually implicate our molecular biology technological knowledge, and we can explore those microorganisms. We can actually map those unknown microbial entity in our environment so that we can understand about their physiology, we can understand about their biochemical activities, we can understand their potential, which can be successfully implemented in our environment. We can improve our livelihood by using their commercial values, They are having the immense potential. Without knowing the microorganism, we won't be able to move on towards the sustainable development of the livelihood. Therefore, we have got some idea related to the exploration of the microorganisms which are present in our environment. As I said, that only we are able to pinpoint on the I mean 3 to 4% of the entire microbial world in this particular universe. But the remaining 95 to 97% of the microbial world is undiscovered. They are not yet been discovered fully. Therefore, we are strategically moving towards that direction in order to explore those microorganisms which are having the hidden potential in our earth. Now, come to know about, or now deviating from that particular microbial ecology dimension to the another aspect, that is the environmental cleanup. And the environmental cleanup is known as the bioremediation, or mitigating the problem, which is basically the environmental pollution. So now today we will be trying to discuss about some of the aspects which are related to bioremediation. So let me show the, I mean, share the screen and wait for a couple of seconds. Well, so I hope that everybody is able to visual, I mean, able to see the screen as I have already shared. So in the particular first thing, and the first and foremost, that the thing I should mention over here, that is the environmental biotechnology. Basically, the environmental biotechnology is a particular concept, or is a particular strategy through which we can solve different problems which are happening in our environment. So basically, we are applying the blazings of biotechnology, the advancement of the biological science, the modern biological science. We can implicate the modern biological science for the sake of our environment, in order to get the sustainable environment, in order to keep our future generation in the positive direction. We can implement the biotechnology, this particular perception, or the particular dimension of research is very important for us. Now, why we people are bothering about the environmental biotechnology? Yes, definitely we should take the preventive measures of environmental pollution. But whenever we have observed that some places or some sphere has already been contaminated or polluted, in that case, how could we get rid of that kind of problem? So we are observing that some of the places which are already occupied by some of the hazardous materials, or some of the chemicals which are unprecedented to that particular environment, which can harm the ecosystem. So we can actually treat those waste materials, and we can actually handle those hazardous waste materials with the help of the technological advancement. So we can actually implicate the microorganisms or different kinds of live forms in order to remediate, or in order to clean up those environments. So we are basically kind of decontaminating, or we are cleaning those environmental sectors. Now also, we can, what we can do is, by, so the thing question sometimes comes, why should we bothering about the environment? We are not actually going to clean the environment, because sometimes we are taking the initiative to remediate the environmental problem, or to clean the environmental problem, but it should be sustainable, right? So one day pollution free, it's not is working, right? Every day we should clean the environment. So this habit or this practice should be actually assimilated in our vein, or in our mind, basically. So for, in order to have the sustainable practice, we should actually take some measures through which we can extract the energy, valuable energy, from the waste material. If we can extract the valuable energy from the waste material, then if we can wisely use the waste material in order to extract the energy, then that would be very much sustainable. That practice would be sustainable because we are gaining something from the waste material. If we are not gaining something from the waste material, so what is the utility of handling the waste? Because we people, it's our thought, we are not going to make it habit so that we can clean environment every day. Whenever we are having a kind of benefit, we are observing, yes, there is a kind of benefit, that's why we can concentrate on that particular field. If we are not finding any benefit, we will all the time we will use to overlook from that particular dimension. So it is important that we should take some strategy, or the biotechnology is a kind of technological advancement which can give us the idea to extract the energy from the waste material, so that our interest to treat the waste material should be kept or should be sustainable. That's why we can actually implicate the knowledge of biotechnology in the field of environmental cleanup. Now, next, what I have been discussing, or what my intent to have a discussion over the period of time, is basically the bioremediation. And here in this context, I must tell you that bioremediation is one kind of process where we can use different life forms in order to decontaminate a particular environment where the environmental pollution is a matter of concern. So we can decontaminate, or we can actually clean up the environment. Those environments are already polluted. Now we can implicate the microorganisms are implicated. Also, we can implicate different other forms of biota or life forms like plant or algae or fungus. We can use different other organisms for the decontamination of the particular environment. So biodegradation or bioremediation, particular this process is very relevant in order to treat the environmental pollutants. Now, sometimes what we observe that two different categories of molecules, organic matters, are present in our environment. Sometimes we observe that some of the places which are contaminated with the molecules which are easily degradable, those molecules are biodegradable molecules. On the other hand, some of the molecules which are not biodegradable, and the molecules are very much resistant towards degradation process. Now, if you implicate microorganism, or microorganisms sometimes fail to degrade those synthetic compounds, and these organic compounds are known as recalcitrant molecules. These recalcitrant molecules are very difficult to be degraded. Now, these kind of molecules are known to us as xenobiotic compounds. And these xenobiotic compounds are produced or are synthesized by human. We people are synthesizing this kind of xenobiotic compounds like pesticides, as I mentioned in the previous class. Like one more thing that is important, once we have been discussing about the microbial degradation, the remediation is basically the cleanup of the pollutant. It means the pollutant or toxic substances should be degraded. After degradation process, the pollutant will be converted into the lesser pollutant. I mean, in the degree of pollution will be less, or the converted molecule or the transformed molecules will be harmless molecule for the society or for the environment. So these microorganisms are creating some of the enzymes in their cells. These enzymes could be the endogenous enzyme, or it could be exogenous enzyme. Whenever the microorganisms are secreting the exogenous enzyme outside of the cell, these enzymes will be secreted to the environment. It means the enzymes will be secreted to the, say for example, the soil, or towards the water environment. Whenever these enzymes are secreted, these enzymes are able to degrade these organic molecules. Why microorganisms? The question comes, why microorganisms are going to degrade, or I mean going to secrete these particular enzymes? Yes, microorganisms are able to secrete the enzymes, and the reason behind, microorganisms are having the genes, and the genes are expressing into the protein or enzyme. Enzymes are protein in nature. We know about this particular fact. So the protein or the enzymes are expressed from the gene. So the gene expression is actually producing the enzymes. And now the enzymes is for the livelihood of the microorganism, because microorganisms sustainability is dependent on the enzymes, because they are carrying out their metabolic activity. In order to sustain in the environment, microorganisms do carry out their metabolic activity. During the metabolic activity, they are producing or secreting enzymes. They need to degrade the molecules in order to survive. In order to survive in the environment, microorganisms require the nutrients like carbon, nitrogen, phosphorus, and different other molecules. Now, whenever the microorganisms are observing that the nitrogenous molecules or carbonaceous molecules are present in our neighboring environment, in order to degrade these particular molecules, they are secreting the enzymes so that the large complex organic molecules can be broken down and the smaller form or the small molecules can be transformed or converted, and the small molecules can be imported inside the bacterial cell or inside the microbial cell. So the microbial respiration or the microbial metabolic activities can be operated. So the co-metabolism is a particular terminology comes here because the microorganisms are producing the enzymes in order to utilize the nutrients which are present, as well as the microorganisms are extracting or expressing the enzymes which are helping in order to degrade the different kinds of pollutants. Now have a look into this particular chart. Here in this case, if we treat the pollutant as inorganic and organic pollutant. Now, if you classify the pollutant as inorganic, and also we can classify it as organic pollutants, sometimes we often find that the organic pollutants can be classified as solid organic compound, liquid organic compound, or gaseous organic compound. So gaseous organic compounds are basically volatile in nature. Liquid molecules are present in a solubilized form. Solid molecules are basically they are existing in a solid state. Now the pollutants are basically in our environment. There is a kind of three-dimensional, as a kind of triangular interaction that the pollutants, environment, and organism. Basically, organisms are residing in our environment, and environment is polluted with pollutants. Now, this particular triangular shape is the interaction in between pollutants, environment, pollutant, microorganism, and organism and environment. So, this kind of interactions are going on continuously in our environment. If you talk about the organisms which are present in our environment, what are the different kinds of organisms that is known to everybody? We all know that the organisms could be the prokaryotes, it could be the microbial populations like prokaryotic microorganisms, as well as the eukaryotic microorganisms like fungi or eukaryotic microorganisms, as well as we can consider also the plants, the different kinds of enzymes which are produced by the different kinds of living cells. So these are the derived from these are the source of the organisms or the living organisms, the microorganisms, plants, and enzymes. These are potential, or potential things which can actually treat the pollutant from the environment. Now, if you talk about the environment, different biospheres are there, and as a whole, we can classify different components like water, soil, and air. These three different components are present. Now, whenever we are talking about the pollutant can be degraded or cleaned up by the microorganisms or plants or enzymes from the soil, water, and in water and air environmental and condition. So now the thing is that what are the different aspects which are relevant in discussing during the discussion of bioremediation? Whenever we're talking about the bioremediation, the different terminologies are very relevant, and it comes in our mind. These are coming in our mind. Basically, these terminologies like environment is very important. Whenever we're talking about bioremediation, we are talking about bioremediation means that we are having the remedy with the help of biological substances. So the organisms or living things are important. Now, whenever we're talking about the bioremediation, it indicates that the pollutant free or the pollutant should be free from the environment. That's why pollutants, this terminology comes in our mind, right? So bioremediation process can be carried out by different ways. We can implement different strategies. We can implement the plant. So whenever we are implementing the plant community, it's known as the phytoremediation, as I said earlier. Phytoremediation, whenever we're talking about the without living biota, we can also treat the waste, or we can actually treat the different kinds of contaminants. We can use enzyme. So enzyme immobilization is another process where we can actually embed enzyme molecules into a particular substance, into a particular solid support, and we can treat the pollutant. we can actually implement biosurfactant also. So this particular terminology will come, and we will be discussing about the biosurfactant. What are the biosurfactant created by different microorganisms, or different organisms are creating biosurfactant, which can exhibit a potential role in order to remediate the environment. We can actually use different kinds of enzymes, and we need to know about the different kind of enzymes which are potential for degrading different kinds of pollutants. So we need to isolate different kinds of enzymes from different microorganisms or different organisms. So different organisms are producing or having different kind of potential. That's why we have to extract or isolate different kind of enzymes. Now we have to take some measures about the techniques or technologies which are related to bioremediation technologies. We can actually immobilize the cell. Cell immobilization is basically we can implement the microbial cell or different other cellular, cells in the different membranes or different other solid substratum, and we can immobilize them in order to get the bioremediation. So the biofilter can be produced by immobilizing the cells. Now, the bioremediation process can be carried out in the direction of metal. We know about the metal, different kinds of metallic compounds are contaminating our environment. So the metal contamination can be remediated by different organisms, starting from the eukaryotic plant. Also, we can implement algae, fungus, and the bacteria. I will show you the different organisms which are involved in metal remediation in the literature. I am having the literature at the end of the discussion. I will be showing that particular literature. Now, what are the different strategies, or what are the different steps we should undertake during the biodegradation or bioremediation study? Basically, we can, whenever we are talking about the bioremediation study, say for example you are collecting or you have collected a pollutant like plastic pollutant in from the waste disposal garbage land. Now, after collecting the waste disposal, after collecting the plastic substances from the waste disposal land, what you are doing, you take it, I say for example, you have brought it in a laboratory. After taking it in your laboratory, you can actually culture a particular type of microorganism which is having the ability to degrade the plastic. So important thing is that all the microorganisms which are present in our soil is not going to degrade the plastic or any kind of recalcitrant molecules. So there must be some of the population or of particular microorganism which is having the potential to degrade the plastic, or which is having the degradation ability of polyaromatic hydrocarbon, or which is having the ability to degrade the polycyclic biphenyls, or which might also be relevant in degrading the trinitrotoluene, which is an explosive. So these are the different organic molecules can be specifically degraded by a particular type of organism. So we have to isolate, we have to fish out, or we have to pinpoint on that particular microorganism. We have to collect that particular microorganism from the soil environment. So the strategy is to isolate the microorganism from the soil environment. After isolation, you have to purify the microorganism, so that the single microorganism, you don't need 2 or 3 different microbial population as a kind of consortia. What you are doing, you are just taking the important or relevant microorganism which is having the ability to degrade that particular material, like X material degrading bacteria. So we are just trying to focus on that X degrading bacteria, not we are talking about other bacteria which is not relevant in degrading, or which is unable to degrade that X material. So we will be trying to focus on that X degrading bacteria. So we are trying to isolate that particular bacteria, and we are purifying that particular bacteria. What we are doing is, after purification of that particular bacteria, the next step or the strategy will be like we have to identify the organism, whether the organism is bacteria, or the organism is fungus, or it is a kind of different other organism or not. So say for example, you have identified the bacteria as identify the microorganism as bacteria or fungus. That's fine. You identified that it is bacteria, or either it is bacteria or it is fungus. But you have to know what kind of bacteria it is, or what kind of fungus it is. whether the bacteria is E. coli, or it is Bacillus subtilis, or the fungus is Rhizopus oryza, or the fungus is Penicillium notatum. So these are the different classes of microbes or different kinds of categories of microbes. We can actually identify the microorganism at species level, not only genus level. We have to identify them at species level, and also sometimes we are isolating the organism and we name them by denoting them as the strain actually. So up to strain, we have to classify the organism in the taxonomical level. After that, we have to optimize, or we have to understand whether the microorganism is potential in order to degrade that X chemical or not. So we have to optimize the rate of degradation of that particular molecule in the laboratory condition. So we make the laboratory condition or a kind of setup where we can actually implement the microorganism. We can apply the microorganism, or in a small scale, we can see how the microorganisms are going to degrade the particular molecule or recalcitrant molecule. So we have to study the degradation kinetics. So the time dependent degradation kinetics will give us an idea about the fact that yes, the microorganism is growing and the microorganism as well as is degrading the particular organic molecule. So we have to assay whether the organic molecule is gradually decreasing in the environment or not. At the same time, the molecules, I mean harmful hazard or harmful effect of that particular toxic substance is diminishing in our environment. As a result, the environment is going to be cleaned up and the environment is purifying. Basically, it's a kind of purification process, purification of the environment by the microorganisms. So we are optimizing the rate of degradation of that particular microorganism. Say, for example, you are having two different microorganisms. One microorganism can potentially degrade that particular recalcitrant molecule in a faster way. Another microorganism which is unable to degrade, or which is not actually degrading that particular substance in the faster way. The rate of degradation is very poor. So the rate of degradation, if the rate of degradation is very minimum, so we can actually undermine that particular organism, or we can actually avoid those kind of microorganisms that are not valuable, that are not commercially, usable. We are unable to convince the people, the commercial people. They are not going to be convinced by your organism that the organism is very poor. So you are having the potential organism, so why not you are going to use that potential organism instead of using the potential organism? You cannot use these organisms which are not having the industrial value. So your organism will be will be important if it is having the industrial value. So you can implement this kind of microorganism by optimizing. Otherwise, we are not going to implement the microorganism in industrial level. After optimizing their degradation in the pilot scale, we have to go further for the industrial scale, and we have to scale up in this way. So the biodegradation efficiency determination is very important in this particular case. So we know that the biodegradation, rate of biodegradation, and the biodegradation authenticity has to be evaluated. Now, the thing is that once we understand that the microorganism is able to produce a particular enzyme which is specifically can degrade the molecule, like if you talk about the cellulose is a particular organic compound. If you talk about the enzyme which is relevant for degrading the cellulose, you can consider the cellulase enzyme. Cellulase enzyme is able to degrade the cellulose. It's known to everybody, right? So if you can purify the cellulase enzyme from the microorganism, then it will be very, it will be a good approach, or it will be a very valuable asset for you. You have got, or you have you have isolated of potential, or the particular enzyme, that is cellulase, can degrade the particular cellulosic material. Now the cellulosic material can be degraded very easily. Yes, of course. Now with the help of this particular enzyme, what you can do is, after extracting this enzyme, you can immobilize this enzyme. Whenever you don't have that particular bacteria, you can use the enzyme in immobilized form. In the filter, in the any kind of solid substratum, you can immobilize the cellulase enzyme, and after these you can implement this kind of granules or this kind of solid substratum for treating the cellulose waste material. So whenever we are talking about the agricultural waste material like lignin or lignocellulosic material, you can use the cellulase enzyme, and you can use this immobilized enzyme for the degradation of the agro waste chemicals, agro waste materials. Now, this enzyme identification and characterization is also important so that we can convince the people, yes, my enzyme or my purified enzyme is potential enzyme and can specifically degrade that particular molecule. So it becomes very convincing for us. Yes. So the thing is that, as I said, that the different aspects of the enzymes and utility of the enzymes, we can actually go for cell or enzyme immobilization. Basically, we can immobilize enzymes or cells in a particular material. We can actually adhere the cells or enzymes either physically or chemically. We can actually retain the enzymes in the particular material, so that the enzyme's activity or the catalytic activity of the enzyme should not be hampered. You have to remember one thing, that the enzyme activity should not be hampered. If the enzyme activity is hampered, then it is a futile exercise. It is not going to be working, right? Because your objective is to treat something, is to treat or is to degrade something, some molecule. If the enzyme activity is lost by immobilization process, during the immobilization process, then your objective would not be fulfilled, right? So in enzyme immobilization process can be carried out by different ways. Like, these are the conventional ways, like carrier by binding methods are there, cross-linking methods, entrapping methods. These are the different various methods as given in this following chart. Now, in carrier binding method, there are different four different kinds of methods are there. Entrapping methods are also including of lattice method, microcapsule formation, membrane method, and reverse micelle formation method. These are the various aspects through which we can actually immobilize the cells or enzymes in the solid substrate. I mean, the physically or chemically, we can do that, right? Now, the materials we can use for the enzyme immobilization process, we can use different kinds of polysaccharides. These are the examples of different polysaccharides we can use for enzyme immobilization process. We can use proteins like gelatin, albumin. These are the proteins which can be implemented successfully for this particular objective. We can also use different kinds of synthetic polymers which can be synthesized, like polystyrene derivatives, polyurethane. These are known compounds. Also, we can use the different kinds of inorganic materials that are glass and ceramic, that is known to use ceramic, and magnetite also used for the inorganic materials. These are the different materials are used. Also, a few materials are also there. So we are just trying to give you the glimpse of the different materials. So also we can talk about the covalent binding methods. As I said, some of the methods are common. One of the important method is cyanogen bromide method, is important in order to make the covalent bonding or covalent binding of the particular enzyme immobilization process is important. Also, there are acid azide derivative method is also important. So in addition to these, various methods are also working in order to immobilize the cells. If you talk about the cross-linking method, there are various methods are also there in order to make the cross-linking between polymer and the particular enzyme, and you can make the chain of polymer, and you can use, say, for example, glutaraldehyde is also important in order to react with the enzyme cross-linking process. So these are the various aspects or ways through which you can make this kind of trapping. Also, we can talk about the entrapping, which is a very popular method. Entrapping, it means that in a lattice, you can actually embed these enzymes in this way. So in this kind of, this is circle and this is the square shaped, these are the lattice, and within the lattice, you can actually entrap the particular molecules. These are the enzymes, and you can make this lattice with the help of different kinds of molecules. Like these are the polymer, like acrylamide, alginate is found from the particular algae. Vinyl alcohol polymers are produced from organic synthetic process. You can also talk about the microcapsule. These are the microcapsules, droplets. You can prepare the microcapsule droplet, and you can actually entrap these molecules, these enzymes inside the microcapsule. So these are the traditional, or these are the different approaches through which we can actually entrap the different molecules. Now these are the different ways or methods through which you can entrap, or you can actually embed different kinds of enzymes or cells for in mobilization process. Now talk to move on towards the another dimension, that is phytoremediation process. As I said, that phytoremediation is one of the important aspect of bioremediation process, where we can use the plant community in order to accumulate, in order to remove, or in order to convert the pollutants from our environment. Now, in this direction, I mentioned in the previous class, I have told you about the various aspects of phytoremediation, that is phytostabilization, phytovolatilization, phytostimulation, phytotransformation, and phytoextractions. I have described all these things in the previous class. So the phytoremediation is basically the you know all about the particular microorganisms or the microbial consortia present in the rhizosphere zone of the plant. Now, the microorganisms are in a symbiosis or symbiotic relation. They are actually promoting the growth of the plant community in the particular soil environment. Whenever the plant communities are growing in the soil environment, once the the plant community is finding the particular different kinds of pollutants which are present in the soil environment, they are potential enough in order to accumulate these particular different kinds of pollutants. It could be like the metal also. So the pollutant stabilization or pollutant accumulation process can be carried out. So various processes, as I said, that stabilization or degradation of the pollutants, or the extraction or the extraction of the pollutants by the plant root can also be done. So the phytostimulation or the degradation of the particular molecules or the pollutant can be carried out by the mutualistic interaction between the plant and the rhizosphere bacteria. So these bacteria and the mutualistic interaction is going to improve the rate of the remediation to some extent. Now, also we can also have a look into the fact that there are different plant species or plant community have already been discovered that are showing the enormous potential in order to remediate different kinds of contaminants. For example, the heavy metals. So the heavy metal accumulation can be done by various kinds of plant species, and it has been found that more than 400 plant species are already found. Those are able to potentially decontaminate or remediate the heavy metals. I will be showing you a literature, a paper, as I already mentioned in the very end of this particular discussion about the literature, and these will be presenting you about the microorganisms or the plant community or the fungus which will be remediating different kinds of heavy metals. Now talk about the root and the root exudates of these plants. They are playing very important role in remediation or in phytoremediation, more precisely I would say, as it is going to activate the surrounding microorganism. Basically, the microbial populations are in a mutualistic interaction. Whenever the microorganisms in the rhizosphere zone are getting the plant root exudates, they are getting activated in order to remediate in order to degrade those pollutants which are present in the rhizosphere zone. So the rate of remediation is going to be increased by the microbial population present in the in the rhizosphere zone. So it's a kind of very good mutualistic interaction, so that this mutualistic symbiosis relationship is very important in order to have a enhanced bioremediation potential. So also we talk about the blessings of microorganisms are some of the microorganisms which are producing insecticides, or some of the microorganisms which are going to produce the phytohormones which are actually improving the growth of the plant, so that the plants can promote their growth as well as they can secrete the plant exudates, and they can as a whole it has been found that that kind of interaction going to be very stabilized, and the rate of the remediation will be very fast. Now, talk about the remediation of metal. As I said, that metal things are metal can be immobilized or metal can be solubilized. If we are going to solubilize the metal, the process will be regarded as the leaching process by the help of the some potential living organisms. Those are known to us as bioleaching process. Bioleaching process is basically the solubilization process. Here in this case, we can actually extract or we can actually solubilize the metals, and the bioleaching process can be carried out with the help of some of the organic acids, which are it could be acetoacetic acid, butyric acid. These are the potential organic acids, can be potential one in order to bioleaching activity. Also, we can talk about the siderophores, which are the molecules can actually chelate the iron. So it is iron chelating molecules, or siderophores has gained attention nowadays. It can be implemented successfully in order to chelate the iron from the environment. Also, you can talk about the root exudates, or I have already mentioned, that are also relevant in order to solubilize the metals present in the rhizosphere zone. Also, if you talk about the metal immobilization process, the metal immobilization process can be carried out by two different ways. One is either precipitation ways. So you can talk about the important bacterial, community, which are actually going to, these are anaerobic bacteria. They are going to produce the H2S producing bacteria, or sulfate reducing bacteria. So the sulfate reducing bacteria can precipitate the different metals, and the organisms which are creating or secreting the siderophores, or the organisms which are able to use the metal as terminal electron acceptors, they are actually going to reduce the metal. So the metal reduction process is also important. If you talk about the complex formation process, bioaccumulation or bioabsorption process, now the bioabsorption process is basically a kind of physical phenomena where the living organisms are secreting some of the biological materials which are secretory, and the biological materials are having the potential to adhere. These are the polymers. These are having the kind of gelatinous material or kind of adherence property. These kind of materials are going to adhere with the different kinds of metals, ions, and make a complex. So polysaccharide materials like exopolysaccharides, EPS, exopolymeric substances created by the biofilm. So one kind of interaction in the micro environment, the biofilm is the microbial, the interaction of the microorganism in the microhabitat, so that different kinds of microorganisms can form a kind of biological film, and they can actually produce or secrete exopolymeric substances which are having potential role in order to make the complex with the heavy metals. Now, also, we can talk about the lipoproteins. Lipoproteins are also important things which are having the ability to make the complex with the metals. Now, talk about the other things which are relevant, different kind of things which are relevant in order to degrade different kinds of pollutants. Like if you talk about the fuel oils like gasolines, ketones, alcohol, monocyclic aromatics, or bicyclic aromatic compounds. These are the different compounds can be degraded very easily by the different kinds of materials. These are the known contaminants, or different kinds of pollutants can be degraded with the help of different kind of living organisms. So these are the some examples of siderophores that I told. Siderophores are secreted by the biological biological organisms like microorganism. For example, Pseudomonas is able to secrete the siderophore molecules. So these siderophore molecules are having the chelating ability, iron chelating ability. Now I'm going to talk about the another important dimension which is very important, and it is biosurfactant, of course. Now the biosurfactant is a kind of chemical compound which is having the property like hydrophobic property, or it is having the hydrophilic property. Like hydrophobic and hydrophilic, you all know about these thing is the hydrophobic property. It means it is having the non-polar activity or non-polar interactive ability, or hydrophilic activity. Whenever you are talking about, it indicates that the particular the water loving activity, or the polar molecules which are having the ability to interact with the water molecule, but hydrophobic molecules are not able to interact with the water molecule. So they are basically those molecules are having both the hydrophobic and hydrophilic property. They are popularly known as amphipathic molecules because they are having both the two ways, like phospholipid. In case of phospholipid, you can see the hydrophobic tail as well as hydrophilic head. So hydrophobic head and hydrophobic tail and hydrophilic head. Glycerol is the hydrophobic. It is having the hydrophobic head, and the fatty acids are the non-polar chain. So these are the examples of amphipathic molecules. Now, in this particular context, I would say that, of course, these biosurfactants are also those chemicals which are amphipathic molecules. Now, the biosurfactants are, since the name suggests that bio indicates the surfactants are produced by the biological substances, which are the bacteria, like cyanobacteria or blue-green algae, fungi, yeast. These are the different kinds of organisms which are producing different kind of biosurfactants, which are having the potential role in order to extract or in order to attach or adhere with the different kinds of organic compounds or different kinds of metallic compounds. These are having the adherence property. Now, the biosurfactants, like, you can talk about the glycolipids, you can talk about the lipopeptides, phospholipids, glycoproteins, polymeric biosurfactants. These are the different kinds of molecules, organic molecules can be treated as biosurfactants. Now, the chemical structure of biosurfactants, as I said, the glucolipid or a structure of the microorganism derived glucolipid structure. These are the complex structure of the glucolipid and can be derived from the microorganisms, and these are the examples of biosurfactants. So why we people are bothering about the biosurfactants? What is the important role of biosurfactant? Basically, the biosurfactants are a particular kind of compound which is having the ability to increase the availability of hydrophobic compounds in a particular solution. It is going to interact with the hydrophobic molecules, and it is going to make the availability of the hydrophobic compounds. Now, also, it has been found that the nutrient storage molecules are also showing this kind of biosurfactant properties. Now, you can also talk about the harmful compound that can be effluxed from the cell. So harmful compounds, once get inside the cell, these particular molecules can be get out of the cell. It is a kind of detoxification of the cell. So the biological cells can be detoxified by the biosurfactant. Whenever the organisms are producing the biosurfactant, or plant community or any kind of microbial cells, they are producing the biosurfactant, they can actually flux the harmful compounds from the cell, so that their self-defense mechanism is like biosurfactant. Now, we can also talk about the extracellular or intracellular interactions, such as quorum sensing and biofilm formation. Remember one thing, that quorum sensing and biofilm formation is a very emerging field of research nowadays. Now, the quorum sensing and biofilm formation, this is basically, it's a kind of quorum sensing is a kind of signaling molecule, is a kind of signaling process where different signaling molecules like acyl homoserine lactone is one kind of signaling molecule which is being secreted by one particular type of bacteria. And after secreting this kind of signaling molecule, if the population density reaches to a maximum level, to a threshold level, then the population will form the kind of biofilm. They will actually conquer in order to make themselves sustainable in prolonged period of time. For the prolonged period of time, they will be sustainable for prolonged period of time, like they can withstand the environmental stress. Like these microorganisms, whenever they are forming the biofilm after quorum sensing process, they can withstand different kind of hurdles, they can withstand different kind of harsh environment. Like if you put antibiotic to the microorganisms, those are making the biofilm, they are not being treated by, or their microorganisms are not going to be destroyed by the antibiotic. If you treat microorganisms with the help of antibiotic, the antibiotic won't be able to perform its activity. So the antibiotic will fail to treat the disease. So whenever the microorganisms are forming biofilm in our human, in case of human infection, it has been found that the chronic human infection has been created, are usually created by the biofilm formation. Those microorganisms are the potential microorganisms and can create the biofilm. They are very much dangerous for us. They are actually creating diseases by biofilm formation. Whenever the biofilm formation are occurred in the human body, then if you treat the antibiotic to disrupt the biofilm will not going to be responded. They are not going to be responding in presence of the antibiotic. So the antibiotic is not going to work well. So your antibiotic will fail to combat with this kind of microorganism, because the microorganisms has already made the biofilm formation, or the complex creature, or the complex kind of interaction has been formed. So it's a kind of hard shell or hard network shell is formed outside of the microbial community, and this shell is not being penetrated very easily by the antibiotic, or any kind of drug is failed to penetrate this kind of biofilm. So biofilm research is nowadays is a very important aspect, and we should take a look into this particular domain also. Now, biosurfactant in bioremediation we can use, obviously. So those are the water soluble substrate molecules, poorly water substrate molecule, or the hydrophobic molecules can be pseudo solubilized by the help of the biosurfactant, so that it could be bioavailable or it could be accessible by the microorganisms. Also, we can actually reduce the absorption of non-polar pollutants to the surface of soil particles. So these are the examples of biosurfactant. And also we can talk about the bioremediation technique. Whenever we are treating the different kinds of solid or different kind of materials which are the particular pollutant, we can actually strategically we can treat the pollutants by either by in situ remediation or ex situ remediation. Whenever we are talking about the in situ or ex situ remediation, we must know two important strategies are there. One is the biostimulation process, another one is the bioaugmentation process. Now the biostimulation process is basically those processes which are actually the process which are going to stimulate the biodegradation rate, and we are stimulating the biodegradation rate with the help of optimizing or manipulating the physical parameters or chemical environment. Physical or chemical environmental manipulation is going to improve the rate of the biodegradation or rate of the bioremediation process, is known as biostimulation process. So we are actually stimulating the biodegradation process with the help of manipulating the environmental condition. But in case of bioaugmentation process, you are incorporating or you are just introducing the microorganism, potential microorganism in the environment, so that you can improve the rate of the degradation. So in case of bioaugmentation, you are just augmenting or you are increasing the rate of the biodegradation after implementing the microorganism. But in case of biostimulation, you are just putting or you are manipulating the environmental condition in order to improve the rate of the degradation. So this is the difference between biostimulation and bioaugmentation. Now, these are the examples of bioremediation, biostimulation process in bioslurry reactor or biopiling process. Biopiling process is a known process where you can actually, make the system aerated. You are just providing air. Circulation of air is giving the ambient condition is a kind of, it is giving the pace in order to, it is giving the stimulus in order to improve the rate of biodegradation. Biopiling is a process through which you can actually aerate the system. You can actually incorporate the oxygen. So you are just oxygenating the system in order to upgrade the rate of the degradation. So this is one process, and ex situ remediation process, where you can actually biostimulate the particular degradation process. You can do this. Now, also, if you talk about the land farming process, is very important, and nowadays it is important for us, because land farming is a process where you can actually implement the bioremediation process. You can actually talk about, or you can actually consider different waste material and you can actually use the biological waste or the biodegradable waste material in order to use this waste material in the land or agricultural land. You can actually make the compost, which is known as biocomposting. So biocomposting is a process through which you can actually degrade the solid waste material. After degradation process, you can convert the waste material into the organic manure. You can convert the waste material into the, I mean, biologically available fertilizer. So these organic manure or fertilizer or compost can be used for land farming process. For the agricultural activities, you can implement this kind of waste material. So bioremediation process is coupled with land farming process. So the bioremediation process is now coupled for agricultural activities. So it is a kind of value added, I mean, extraction of waste material into the value added products. So it's a kind of value added product extraction or value added product production process is relevant in this particular domain or direction, right? So now I just want to let you know one thing, that there is water and gas bioremediation process. So whenever we are talking about the biofiltration process, is a very important process where you can use different things. Like if you are talking about the water or gaseous substances, whenever it is polluted, say for example water pollution, water is getting polluted or gaseous molecule in the air is getting polluted with the different kinds of contaminants. If you are observing this kind of pollutants, then you can actually use the filtration system. So this kind of filtration system is known as biological filters, or biological filtration system is a process where you can actually use microorganism in the inert material in order to degrade the organic pollutants which are present in air either, or in water, or in gaseous phase. So this is a kind of remediation, bioremediation process. You can implement bioscrubber, you can implement biotrickling filter, you can use the carbon filters or sand filters. So these are the different filters you can use. The example of the different kinds of filters you can use for treating either water or for treating the air. Also you can treat different gaseous molecules which are emission from the different industrial hubs, or we can treat the wastewater treatment plant by using the trickling filter, which is going to treat the wastewater. And also we can use the biological filter, bioscrubber, in order to purify the air also. So these are the different aspects we can consider, and we can remember all these things. Like we can actually use the carbon filters or the slow sand filters for purification of water samples or water materials. Here carbon filters can be used in water purification system also. So these are the different aspects, or these are the different various aspects related to bioremediation and biodegradation. Now I'm going to show you one example or one research paper or on a review paper which is going to show you about the remediation or phytoremediation. Better I should say that phytoremediation, or the heavy metal remediation process, which can be carried out by different kind of organisms. I'm going to show you the literature which is a kind of example, and we can actually have a look into this particular metal remediation literature. So just a minute, wait for a couple of seconds. Let me open the research, the article. Well, so I'm going to show you the article which is known as. So basically this review article is going to tell us about the, I hope everybody can see this, right? Is it visible to everybody or not?
Yes, sir, it's visible.
Okay, thank you so much. So this is the review article which is going to show us that the bioremediation, the opportunity of the biotechnology is going to give us the opportunity to remediate the heavy metal. So heavy metal pollution is a growing concern, and bioremediation is the way through which we can actually remediate the heavy metal pollution. So heavy metal remediation process is a very important aspect for the current days. Now, if you talk about the heavy metal remediation process, so what are the different options of heavy metal remediations that I'm going to show you in this particular literature? I will share this literature. If you see this literature, you will find that the pollution can be of two types. Two different kind of sources are there. Like here in this particular literature, it is showing that the heavy metal pollution can be anthropogenic pollution. It means the human activities, we are doing the pollution or we are creating the pollution. That's why it is known as anthropogenic activities. Yes, Shopna Mondol has raised hand. Please, Shopna, will you please ask some question?
सर ये मतलब बिल्डर दिखाई दे रहा है सर.
Yeah, what is? Excuse.
सर হ্যালো
Yeah, I can hear you.
স্যার ক্লিয়ার দেখা যাচ্ছে না, ছোট ছোট দেখা যাচ্ছে স্যার.
Okay, I'm going to magnify it. Okay, thank you. If it is visible now, let me know this whether it is visible
Yes
or not.
sir. Yes
Now
sir.
is it fine, right? Okay. Thank you. So see this particular diagram here. It is showing that the source of the heavy metal could be anthropogenic source or natural source. So the natural sources are like mineral weathering, soil erosion, volcanic activities, forest fires, vegetation releasing particles. These are the natural sources of heavy metals. On the other hand, anthropogenic sources, it means the civilization or we people are creating this kind of heavy metal pollution. These are the industrial sector. These are the different kind of emissions are coming from different processes or different activities. We are applying pesticides, fertilizer, and heavy metals are coming from these sources. Ore mining, electronic waste, wood preservatives, application of wastewater and municipal sewage sludge, and personal care products, cosmetic medicines. These are the sources of heavy metals from anthropogenic activities. It means we are actually creating this kind of waste material. And natural origins are like mineral, as I said earlier, right? So these are the different sources of heavy metal. Now, what are the different kinds of heavy metals are there? Like different heavy metals, if you can see list in this list, it is showing that arsenic, cadmium, chromium, copper, these are the different heavy metals. And these heavy metal sources are like textile mill, effluent, electroplating industries, or you can see that different kinds of particular sectors are polluted. Now, river Ganga is heavily polluted. You know about this. The groundwater quality can be contaminated, or the groundwater quality can be degraded by this kind of heavy metals. It is also observed in our state or in our North 24 Parganas. It has been found that the groundwater is heavily contaminated by different metals. Now, the water supply, it has to be monitored whether the water supply is contaminated with heavy metals or not. Irrigation, different other sources are also contaminated by heavy metals. So this is important to be noted over here. Now, if you talk about the different, processes, different aspects that are going to tell us how can we implement our strategies or how can we implement our knowledge in order to why should we bother about these heavy metals? As you want to most of the people are interested to know about the what kind of toxicity or what kind of health hazards are created by the heavy metals. So in this particular picture or in this particular list, you can have or you can see the different adverse effect associated to arsenic, cadmium, and other heavy metals also. So it is written in this particular chair what kind of diseases or what kind of toxicities are created by these different kinds of heavy metals, which are lead to cause cellular level damage, or it can create different kind of DNA damage also, which might have the carcinogenic effect in our health. Sometimes it has been found that few chemicals or few heavy metals are creating neurodegenerative diseases also, DNA damage, and it can create different kinds of neurotoxicity also. So different kinds of broad spectrum activities of the heavy metals are there in cellular level. So we should get rid of these heavy metal pollution, because heavy metal accumulation in human or in animal can be dangerous or can be deleterious for us, for our society. So we can have a look into this particular the different aspects of different kind of consequences, adverse consequences created by different kind of heavy metals. Now, whenever we are talking, as I already mentioned about in situ and ex situ bioremediation process, it is mentioned over here. In situ bioremediation, there are different processes of in situ bioremediation as well as ex situ bioremediation are also there. So we can implement ex situ or in situ bioremediation process in order to, combat with this kind of great challenge of heavy metal remediation. So whenever we're talking about bioremediation, we can implement microorganism in order to remediate the environment. So what are the different kinds of microorganisms we are actually using? So we can actually use different kind of microorganisms, living or non-living substances are also used for the bioremediation. So for bioabsorption process, in order to absorb the heavy metals from the environment, we can use different kind of waste materials like agricultural residues, algae, bacteria, fungi, industrial waste materials or other things. These are used for the absorption of heavy metals. So these are the absorbent. These are known as absorbent. So examples of I'm taking just 5 minutes, not more than that. Okay, just sorry for the delay. So the examples are like rice, wheat straw. These are the agricultural waste material. And you know about the fungus, different kinds of potential fungus which are observed in the water bodies are very potential in order to remediate or absorb the particular heavy metals. The bacteria are also there, potential can be the good absorbent. So fungus are also there. Industrial wastes can be used for the absorption process. Now, the thing is that in case of bioremediation process, we observe that different bacterial species are there, which are having the absorption ability to absorb different kinds of heavy metals. These are the list of microorganisms or the bacteria specifically can absorb the heavy metals, different heavy metals potentially. It has already been, discovered or explained very well that these are the microorganisms can absorb the heavy metals. Now, also together with this, we can also talk about the algal species. I hope everybody can see this. Algal species are also there, with different algal species which are potential in order to absorb the heavy metals also. In this direction, I must mention about the algae, bacteria. In addition to these, I should not undermine the contribution which comes from the fungal species, which are also potential in order to remediate or in order to absorb different kinds of heavy metals from the environment. So these are the different fungal species. As you can see in the list, so many fungal species have been discovered. They are having the potential role in order to absorb the different kinds of heavy metals from the environment. So these are the examples of the particular microorganisms which are potential in order to absorb or remediate the heavy metals from our environment. So this literature will be helpful for you all in order to study well. So I will be sharing these particular literature to you all if you definitely you require it for the your study purpose. So I hope that you have got some idea related to the today's presentation. So I would now time has come. Now I am just going to ask you whether you are having any kind of question related to this particular discussion. So will you have would you like to ask any question related to this particular topic? So if you are having any question, you can ask me. So do you have any question? So without, I need attendance. Please don't go away, right? Okay, I'm going to take the attendance. So Shopna Mondol is present, right? Shopna Mondol is present.
Yes, sir.
I'm taking the attendance. Yes. Shopna Mondol is present. Aporna
Yes, sir.
Chaudhary is present.
Yes, sir.
Aporna Chaudhary. Yes, you are present. Right. Aporna Chaudhary is present. In addition, I am observing that Supriyo Kumar Bose, Mr. Supriyo Kumar Bose is also
Yes,
present.
sir. Yes,
Yeah. Tirtho Pratim Shaha
Yes,
is
sir.
also there, right?
Yes.
Yeah. And Tonmoy Boshak. Mr. Tonmoy Boshak is also there. Tonmoy Boshak.
Yes, sir.
Yes, I can. Mr. Tonmoy Boshak is also present. This is the first class, I guess, right? Mr. Tonmoy Boshak.
Yes, sir.
Okay, I called Shopna Mondol, Aporna Chaudhary, Supriyo Kumar Bose, Tanmoy Boshak, Tirthopatim Saha. In addition, I am observing one more name.
Yes, sir.
Is
This
it
is Nutan
Nuton
here.
K? Will you please tell me your full name?
Nutan Kumari,
Good name?
sir. Yes, sir.
Nutan Kumari. Okay, okay. Thank you so much for. I mean, wait just a minute. Nutan Kumari. Nutan Kumari. So, Miss Nutan Kumari, where are you from?
Actually, sir, I'm from border of Jharkhand. Currently, I'm in Hyderabad.
Okay, currently you are in
Yes,
Hyderabad. You are engaged in job
yes,
because this is the first time I am going to interact with you. That's why I'm asking. You
yes,
are in job, right?
yes, yes, yes. I work as
Where are
a
you?
embryologist.
Okay, okay. So you are a neurologist. I mean, you are, I mean, doctor, medical practitioner,
Yes,
right?
yeah. Actually, I'm embryologist, sir. Embryologist.
Embryologist. Okay, ma'am.
Yes.
Okay. Anyway, it's very nice to meeting with you, and I wish you all, everybody. I wish all of the, I mean, members who are actually present in this particular session. I wish you all the very best for your coming examination of coursework. So this is the first time I am taking the class of Miss Nutan Kumari, and Tanmoy Boshak is probably this is the first time. So whatever I have been discussing in the last classes, I will share you.
Yes,
I
sir.
mean all this presentation and all these materials with my HOD, and HOD will post it to the relevant WhatsApp group so that you can study it properly. Okay.
Yes, sir. Thank you, sir.
Also, I'm going to write my WhatsApp number and email address if you require any kind of information related to related to the matter of concern. So you can ask me anytime. I will be feeling good if I can reply you on get back to you in any way. So I'm giving you my email address as well as my WhatsApp number so that you can reach me. Uh, we just a minute. I'm writing it in chat box. Okay.
Yes, sir.
I'm writing it chat box, and so this is my email address, and I have also written my this phone number. This is WhatsApp number. You can write in this WhatsApp number if you are having any kind of doubt after the session. So anyway, I have given these details in the chat box, and I hope that you understood today's topic was really interesting. And if you feel any kind of, or if you find any kind of problem related to this, you can definitely talk to me. Or if you are having any problem, you just knock me. I will be there in every possible ways. Okay. Thank you so much for listening to me. Once again, I'm asking if you are having any question, you can raise your question. Do you have any question? Otherwise, we will be ending this session. Is there any question?
No,
No,
sir.
sir.
Okay. If not, then I am going to close this session over here. Thank you so much for joining and have a very nice time ahead. Okay. Welcome. Thank
Okay,
you.
sir.
You're welcome. Most welcome. Have a very nice time ahead. Thank you all for joining.